round of applause for Dr. Manish and invite the rest of the speakers on the stage for the panel discussion. So I hope you have noted your questions so that you can fire them, right? And you have the experts here who talked about front-end, back-end solutions for digitalization, digital transformation, as well as automated control from upstream, downstream. So they have covered, I think, end-to-end in terms of processing. So hopefully that has given you a lot of insights. So who's going to be the first person having the question? Anybody can give them the mic, if you don't mind, in the front. She's standing. Go ahead. Yes.
Panel, settle down.
Yeah.
Hi, this is Sridevi from Syngene. The question is for Janice. Very nice presentation about the PAT and the Raman data. My question is, you showed us nice validation of the Raman results as well as the offline measurements. Now, where is the technology today so that this kind of validation doesn't need to keep happening? So maybe two parts of the question. First question is, how do you need to keep doing this validation for every process that you develop? And secondly, is the technology getting there where it won't be required any longer?
Yeah, thanks for the question. I think data or model validation will always be required. It's just how intense and how frequent you need it to be. Yeah, so for now, whenever we change processes, so it really depends on how robust your data is. So again, more data that you have will make your model more robust. And if your data consists of many different scenarios, that's when you'll be able to have a better data that can fit into different processes. So I think in answer, validation will always be there. It's just how difficult or how simple, yeah.
So just a follow-up, is the validation needed for every process from a similar platform or let's say a similar cell line? If you're going with the same cell line platform, of course your gene express will be different. Do you see the requirement for rigorous validation every time?
Yes, because cell line is the same, but your molecule is different, meaning your production cell line is different. And every production cell line will behave differently. So if you bring this production cell line into your manufacturing where you're going to run the manufacturing similar for like the next 10, 15 years, then you can always use the same. Yeah, and your model will just get even more robust. So different processes, upstream processes of the same clone, you can use the same model. But once you change your clone, things will be different. They will have different product characteristics.
Thank you.
Thank you. Thank you, Sridevi. Anybody else? Okay.
This is Anurag from Syngene.
Can you be a little bit louder if you don't?
This is Anurag from Syngene. This is for Dr. Anurag.
Okay.
This is regarding CADET. So I have two questions. First is the query. Is this CADET is accessible for the industry, open source? I'm asking. Is it available for?
Yes, I think so. Yeah, it is.
Okay. Secondly, how are you going to calibrate this CADET model? What approaches we should have for calibration of this model?
Okay. So basically, the way it works is that CADET is, the way CADET works is that you have to do a defined number of experiments, which the software will use to, find because there are a bunch of constants in the model, right? And the software kind of uses the data to calculate those constants. Then you have a mechanistic model of your chromatography step, right? So I don't know what you mean by calibration, but typically the way we do in our lab is that once you have a model, you would run the model on a bunch of scenarios, say varying feed composition, for example, right? And then you would also do HPLC analysis, which is kind of your reference, right? So using HPLC, I can create the same plots I showed because you can see where each variant is eluting. And then you do your CADET thing, and then you compare. So that, in a way, that is your calibration.
So means we have to generate the data.
Absolutely. But just to give you an idea, based on the couple of maps we have done, we are talking about maybe, I would say, 2 months of work per molecule. Yeah.
Got it. Thank you.
So the data that you generate is in lab scale, correct?
Yeah, the data we are generating is in lab scale. I guess the reason you're asking
Applicability.
this is, yes, when you go to commercial scale, there may be some adjustment to the model that you would need to do, right? But I think that is true for pretty much everything. So even the Raman stuff that Dennis talked about, anytime you're changing a system, it's just that the amount of that adjustment will be different, obviously, will not be that much. But yes, that readjusting would be required.
Thank you. Thank you both. Thanks, Anurag and Anurag. The next question, please. If you have any, or there are two hands raised, one more here.
So this is Nanda. I'm from 𝐶𝐻. So the question is about to Dr. Manish. It's about the software which is being used is an end-to-end process, right? So in terms of that qualification or validation of this software, is the process of validation or qualification, is it as like the other informatics software, or how it is?
Yes.
What I understand from your question, like whether this requires the validation and qualification, it has to go through 𝐈𝐽 and that kind of processes. Of course, that's the essential thing when you're working in a regulated market, and it's taken care. It's what you see here is a cloud-native solution, and
You can hear at the back what he said?
Yeah.
Okay.
Thanks.
Okay. There was, right there.
Hello, I'm Prashant 𝐁𝐢𝐨𝐬𝐢𝐦𝐢𝐥 from Lupin Biotechnology. I have two questions, one for Janice and the second one for Anshuman. And my first question to Janice is, in biopharmaceuticals, many years before we were Raman spectroscopy, fingerprint was part of media lot-to-lot variability. These are very key because once we transfer the media into the bioreactor, we cannot do anything. And we have a very classical growth promotion test or few productivity test which tells us to select the lot and release it for the commercial use. Where are we right now in Raman spectroscopy for media lot-to-lot variability?
I think it's still a good technology to use for media lot-to-lot. Yeah.
Right now, is it available commercially in the market, that product?
The Raman technology, the one that I showed in my slide list, I'm sure there's also others. Yeah.
And second question was to Anshuman sir about, we talk about the data analytics, in biotechnology, seeing the complexity of the processes and the molecules, and so much of back-end integration to QC, analytical R&D, such a complexity. Where are we today in that regard? Because end of the day, when it comes to utilizing the data, to get the productivity from the manufacturing and to add value in the CQA or continuous process verification, where are we? What are the current challenges? And how much years more do we need to stabilize in the industry?
I think the current challenges are still that we are very Excel based. So data collection, data capture is all manual, most of the cases right now. But as we go more electronic, I think it will become easier. But the problem is how we store the data also. Like everyone basically have their own custom models or custom data set that they store, and then data model doesn't match each other, and then we don't have accessibility available. But I think things are moving in the right direction. And most of the time we are learning from cloud technology, which are typically, there is a technology called REST, which is representational transfer of data, which relies on technology like XML or JSON. And that is becoming more and more ubiquitous everywhere. It's also coming down to the plant floor. So once that is coming down more, I think the data transfer becomes much easier, because JSON is, you can read the data file of JSON. It's not like bits and bytes. You can read it like a Excel sheet. It's like a string of data. So it's human readable. So it becomes very easy for us to interpret. So once more and more data technology comes on floor, the data exchange becomes much more easier. right now it's not there. It will take another 5 to 6 years or 10 years to come there.
Yeah, and so I feel that watching the industry, that the awareness or the competency in this field is much required, because having the affinity toward the data is very important. And when we know the application or the end use of the data, then people get more interest in getting into it and handling it, or getting into the demos and applying it. I think that is also one gray area where
Exactly. I think the technical, what we feel is scientists don't have to worry about too much of technical coding. So that's what it is required. So I think a lot of the people like our team and a lot of other people in the industry who understand tech is making it accessible for people to use it like your old Excel spreadsheets. So that's when, or it should be like WhatsApp, so that you can start using the data much more frequently. That's not there right now. So there's a lot of gap between the data that gets generated and their data that's stored in the technical language of IT. So that becomes very different right now. That's where the communication gap is right now. So once we can bridge that using these technologies, which are becoming much more easier to accessible, then I think it will be much more easier for us to have seamless integration from different technologies together into one.
Thanks for the questions. Okay, there's one more gentleman here.
Hello, I'm Andreas. I'm with Sativa. I have a question for Janice regarding the Raman implementation. First part of the question: how transferable are the models from your 3-liter scale model, let's say, to a 2K process? And secondly, can you comment on the ease of use, the implementation of the Raman technology on a daily basis? How far are we using it as easily as a pH electrode?
I'll answer the second question first. So, how easy is it to implement? So, like you say, it's really like a pH NDO probe, right? The only consider, I would say, in terms of ease of use would be the time you spend on building your model. So that is the time where we have a lot of effort to be done in order to build your modeling, your chemometric modeling. So implementation-wise, it's really simple. It's really a plug and play kind of system, but it's a model that takes some time to build. So once you have a very robust model, it's just something that you do daily. Yeah. And the first question was on the sorry.
It was about the scalability.
Yes. Yeah. So it's a model that we built on 3-liter. And we have done it in a larger scale of 50 and a 200 liter. So it's not a bioreactor where you have to do scale up parameters modeling because the scale of the bioreactor. The probe will still be the same. The Raman spectrum will still be the same whether it's in a 3-liter, 50-liter, or 2K or even 20K, because the environment is the same. Yeah. So there's no issue in terms of the scalability. It's more of a validation of the model at different scale. So as long as the cell line is the same, the environment is the same, the data can be transferred.
Thank you very much.
Thank you. There's one question at the back. Person is standing. Oh, there also. Okay, yeah. So if you don't mind, yeah.
Yeah, hi, my name is Mayank. I'm from Biocon. So I have a question for Dr. Tan regarding Raman spectroscopy. So as per your experience, how many batch data is sufficient to build a reliable model, for the chemistries what you have shown?
Yeah, so we have recommended at least 20 batches, but it also depends on how many time points that you can get. So again, this is a source census. So as many data as you get from your offline, that will help to validate and build a model. Yeah.
Okay, thank you. Just one follow-up question. So you talked about the multivariate model. What about the AIML-based model? And for that, do we need more data? Or if you compare between the two, which one do you think is the better approach?
So for us, we're using the PCA and PLS algorithm to do our data processing.
Thank you.
Thank you. Lady behind, you stood up before, if you don't mind asking the question.
I'm Anandita. I'm from Campbell. So I have two questions. One to Dr. Anurag. So I know there is a lot of work being done in developing the continuous bioprocessing in terms of purification and also upstream. where are we thinking about the scalability and subsequently scale down process characterization? Because that's the future where it goes to use it for manufacturing for any biopharmaceuticals.
So, I mean, continuous processing has been around in fact, for pharma, it has been around for decades, right? For biopharma, maybe 5-7 years now. But I think it's, I mean, at least for me, it gained traction when I realized that most of the major manufacturers in the US and Europe are at least, they're actually building plants based on continuous, right? So that means people are putting money in it, right? In India, I've not seen that so far, primarily because obviously it's not a trivial thing to suddenly move from a batch processing facility to a continuous facility. Is a huge investment. So companies are hesitating. But I think if you look at the impact on cost of goods, yes, I think eventually, companies will get there. But I think what is stopping is that from a fundamental basis, you kind of have to build a new plant to do it. Your operational complexity increases manifold, right? So I understand that why companies, like Biocon, like Syngene, hesitate to do that.
Thanks a lot. My second question is to Anshuman. So I just wanted to understand, and I think I've been talking about this a long time before, is that we are talking about data storage, in an integrated manner and all that. But as a user, I would like to see that, the data which is getting generated in manufacturing is actual data. And how can we get a dashboard where we can see the multivariate interaction between the parameters, both in, different stages along with the product quality, so that it can help me in choosing, a space where there may be opportunities for future process improvement also.
I think there's some tools available nowadays. I think our old product for process pad, which is now sold by Merck, does that primarily. They have a multivariate dashboard that you can build, and that integrates data from QC and as well as manufacturing together. You can create models, and you can run it real time and see what's your parameter outliers are. So you can build that NBA models on top of it. It's already built in. It's just plug and play. You don't have to code also. So you can try it out, probably talk to process pad team from Merck. So it's only there.
Thank you.
Thank you. Manish, you want to add something? Yeah? Sure.
Of course, the same thing what you said, data. We are talking about manufacturing side has data, and then you want to correlate it with some other data. And again, although you have data systems, it is creating data symbols. So how you want to connect those things. So what I showed is a typical Spotlight based on dashboards. So in the Spotlight, you can read all that data, pull out that data. For that, before you visualize, you need to aggregate all that data. Aggregate that data, will generate uniform data throughout the process down the line. So whether it's your lab scale or production scale or manufacturing. So doing streamline all this data generation and capture processing of the data, then you can bring it in up and you can visualize it as distribution. So you can have the context between your manufacturing data and then what is happened to it in R&D or scale.
Thank you, Manish. Thank you. There's a question in the front row. And we have only 2 minutes left, so hopefully this is the last question. You can talk to them over lunch.
Maybe Dr. Manish or Dr. Tan can answer this. So we talked about all these technologies, and Raman has always been a technology that has been looked at, even in the small molecules, as a tool for doing analysis on a continuous basis or a real-time basis. But we know that this technology is not specific. Secondly, we should also know the limitations of this technology before we implement. We all talk about the advantages, but in the hindsight, we should also understand what are the limitations before required. So what could be the limitations of these technologies, specifically in the biotechnology space?
So of course, there's always two sides to any technology, the pros and the cons. So for Raman technology, it is in a way generic because it can really measure a lot of things. But whether it's useful and it can be implemented in your plant, it really depends on three things. First, of course, Raman is a soft sensor. It doesn't make any sense of the data. You need a equivalent offline sensor or measurement that can give you. So if you are going to measure cell count, you need to have a cell counter in order to make sense of that. So that's one limitation in order to look at what are the variables that you want to measure. And Raman is an effect of vibration of molecules. So in order to generate enough vibration to get a spectrum, there's also limitation in terms of the amount. So it cannot measure ppm, and this is a very generic kind of concern or limitations for Raman spectrometry. Yeah, and again, the third one is cell culture media, for example, is a lot of nutrients, there's products, there's cell count, there's host DNA, everything is inside. So it's also depending on the environment that you're looking at and how well you can separate. So it's like, how well can you separate your peaks for you to be confident that this spectrum is equivalent to your cell protein, for example. So those are the limitations, but I won't say it's a limitation. It's something that you have to consider when implementing, and how would you want to use it, how you want to apply, and what's the comfortable range that you are able to use Raman as your PAT tool.
So, Dr. Maybe also asked about the validation part. Validation is required for every process, every company, every kind of metrics that.
Thank you, guys. Really appreciate it. Time's up. So if you have more questions, I think it'll be a good conversation over lunch. So really appreciate all of your participation, and give another round of applause for the panel members. Yeah, back to Ashwin.
Thank you all for the insightful discussion. With this panel discussion, we are coming to the end. Now I request Dr. Sampath Kumar for the felicitation. Professor Anurag Rathur. Mr. Anshuman.
All right.
Dr. Tan. Dr. Manish. Now I request Dr. Manish to felicitate Dr. Sampath Kumar.
That's how I feel.
Thank you all for attending this session. I request you all for the lunch break. After this, there is 2 more sessions left. That is the Leadership Conclave and one more session. For the Leadership Conclave, it is based on the novels, future of biologics, and the session is single-use technology in biopharmaceutical. It is around 2 o'clock, so you can have the lunch outside the hall. The moderators and other volunteers are available. They will guide you for the lunch. Thank you.